Hi, dear students. Welcome to statistics class. This is our module 3, topic 1, Introduction to Experimental Designs. Okay, let's define experiment. So, any planned inquiry to discover facts or to improve and disprove results of previous studies and investigations. So, it is also like repeating results of earlier experiments and extends them to more complicated and general situations. It is also an act of conducting a controlled test or investigation. It is also revising accepted conclusions, theories, or laws in the light of the newly discovered facts, or demonstrate the practical applications of such new or revised conclusions. So through experiment, we discover new facts or to correct interpretations. So, to review, statistics, it is a body of knowledge that deals with collecting and summarizing data, just like what we did in our frequency, percentage distribution, and graphs, designing experiments and surveys. We also have the layout, describing the data and the variables, estimating population parameters, testing hypotheses about populations, and studying relationship among variables, which will be able to learn in the next topics. So to review again, the role of statistics in research. As I've said, it is a toolkit for research worker in the planning, analyzing, and interpreting the results of his investigations using methods or techniques that have been proven useful in many fields of inquiry. So it is just like a toolkit. Just for example, you have a different experimental designs. So to meet your specific objectives. So by the use or by the guides of statistics, you will be able to meet the appropriate action or appropriate design for your research objectives or for your problems. Also, we have here statistics in research. For example, we have problems in real life, a formulation of specific problems, and then in our data collection, we have the design of experiments, especially in experimental, in historical, and then sample surveys. It's our data collection, and then our data, we have recorded measurements or we have the prior information. So data examination, we have here the data cleaning and test of prior information. And then data analysis, we have model selection and validation. So we have the hypothesis testing, estimation, prediction, and decision making. So this is the role of statistics in research. So to review again, steps in conducting experiments, we have state the problem, which you will learn later how you do your experiments, and then specify objectives of the study, formulate hypotheses, define the data to collect, statistical analysis of data, determine the experimental technique and design, construct the experimental layout, conduct the experiment and collect the necessary data, analyze the data obtained from the experiment, and then summarize the results, make conclusions and inferences, and then evaluate the whole study in relation to similar investigation, just like what you are doing in your RRL. So, for example, we have a problem. Are natural pesticides more effective than synthetic pesticides? So, our objective would be to determine if natural pesticides are more effective than synthetic pesticides. So, how would we determine or what is the response variable to determine the effectiveness of pesticides? So, we have the response variable is the number of insects killed. So let's define some terms. So experimental design is a set of rules, plans, and courses of action taken during the conduct of experiment. So it also involves the assignment of treatments to the experimental units, just like what we did in our layouting. And experimental design is a thorough understanding of the analysis to be performed once the data become available. That's why prior to your conduct of your study, you already have your experimental design, what experimental treatment structure or what design structure you will use. So it is under the concept of experimental designs. So what is the relationship between statistics and experimental designs? So experiments, remember that it involves probability samples. You are doing your sampling. And then experiments lead to inference or conclusions that must be accompanied by probability statements. And experiments should be designed in accordance with the principles of statistics for the probability statements to be valid. So that's why statistics and experimental designs have the relationship as well in your experimental research. Now, what are the importance of experimental designs? So, of course, if you have experimental designs, it's really pre-planned design. It ensures cost-effective collection of appropriate data. So, it ensures that appropriate and necessary data will be obtained using minimum resources because of planning, and then provides a roadmap for appropriate and valid analysis of data. So it permits an objective and valid analysis of results. And also, it would allow for drawing valid conclusions using statistical inference. So it provides reliable inference leading to reliable conclusions with respect to the stated problem. For example, the yield will increase by 20% with 5% level of significance. So you have your valid conclusions, or especially it is connected with your statistics or probability theory. Now let's define some terms again. So treatment or factor, it is a set of experimental procedures or conditions whose effects are to be measured and compared. So it is from the word itself, treatment or factor, which you want to compare in your study. And then the response variable or the dependent variable is the characteristic used to measure the effect of a treatment. For example, you have the plant height, the number of leaves, the yield, so that is one of the response variables in experimental studies. And then sampling unit, or let's use SU, a portion of the experimental unit on which the response variable is measured and observed. So for example, in the rice field, so rice plant is your sampling unit where you want to measure the plant height. Okay, or for example, the number of leaves. So that is the sampling unit. It's a portion of the experimental unit. And then experimental unit is a unit or group of units to which a single treatment is applied. Okay, just say for example, it is of interest to determine if three species of mice will differ in red blood cell count if given iron supplement in the diet. 3 mice from each species were given a diet with iron supplements. So after 4 weeks, 2 blood samples were collected from each mouse and independently analyzed for red blood cell count. So for example, in this species of mice, I've taken 2 blood samples, and also in this species, I've also taken the 2 blood samples, and also in this species. So what is our experimental unit is the mice, but our response variable is the red blood cell count. So we've taken it from their blood. So our sampling unit is the blood, while our experimental unit is the mice. And also, our treatment or factor is the 3 species of mice. So, to review, our treatment or factor tested is the mice species. And then the treatment levels, we have the 3 species of mice. And then the response variable is the red blood cell count. And an experimental unit is 3 mice from each species. And then the sampling unit is the 2 blood samples collected from each mouse. Another definition. Experimental error. So it is the variation in the observed values of the response variable from experimental units treated alike. So I will say that there is an experimental error. For example, this is the fertilizer 14, 14, 14 NPK. I have given them, given the controlled environment, I have given them the same treatment. However, they have the different yield or different height. So I've treated them the same, but the variation between them or among them is called a experimental error. So it is the failure of 2 experimental units treated alike to yield the same response. Because if you have the same of a controlled experiment, if you have the controlled experiment, if they are treated the same, they should yield the same. However, due to experimental error, they varied. That's why we call this error or variation as experimental error. Another example. Our objective is to determine the effect of varying rates of nitrogen fertilization on the yield of corn. And then our treatment levels or nitrogen rates, we have the 0 kilogram per hectare of nitrogen fertilization and 30 kilogram per hectare and 60 kilogram per hectare. So we have the 3 treatment levels. And then 9 uniform 2 square meter plots were planted with corn at uniform density. So each nitrogen fertilization rate was applied to 3 randomly selected plots. So in other words, we have the 3 replications. And so the experimental error consists of pooled variation among the 3 plots with nitrogen fertilization rates. So I have given them, for example, I have given them the 30 kilogram per hectare. So any variation from them among the 3 plots will be our experimental error. So what are the sources of experimental error? As we know, there is no such thing as perfection. So there will be an error. But what are the sources of experimental error? So we have here the inherent variability of experimental materials, just like what we have in a previous example. It might be they have yielded different height of corn, maybe because the soil is different. So inherent variability of experimental materials, or the seeds that I've used are somewhat different, or they are exposed to something which has a contribution in their growth. And then errors in experimentation. So lack of uniformity in the physical conduct of the experiment. So as I've said, maybe in the field, they have the lack of uniformity. And then error in observations and measurements. Say for example, one student have measured the response variable, and then the other student measured the other field, and then they have used the different ruler, or say for example in a different measurement. So there will be errors. So that is also one contributing factor to the experimental error in your experimental research. And as well as we have environmental error, is the combined effects of all extraneous uncontrollable factors which could affect the response variable, just like environmental factors. The researcher could not control that. So it might have some contribution to the variation of the treatments or the response variable treated alike. So that is one of the sources of experimental error. So as much as possible as a researcher, we want to minimize experimental error. But if ever it's uncontrollable, then at least we have some tolerable experimental error. So these are the sources of experimental error. Another term is the sampling error. So it is the measure of variation among sampling units within an experimental unit. So for example, we have our experimental units are one plot. Okay, so the sampling units are here, for example, one rice here, rice plant, one rice plant here. And then the sampling error would be their difference from each other. They are of the same plot, they are of the same experimental units. However, they have the response variable that are different from each other. So the variance or the variation of these units or of these sampling units are called sampling error. So in other words, it is the failure of two sampling units within an experimental unit to yield the same response. So that is the sampling error. Okay, so for example, we have here the plant height is taken from 3 center plants within each plot. So for example, we have the 9 experimental units. Okay. So, and this is the corn. So we have here the response variable as the plant height, and by guidance or by principles, I will take the 3 samples in the center so that whenever there is an error here in the side of this experimental unit, then these 3 plants will be safe because they are in the center. So the sampling error is the pooled variation among the 3 plants within each plot. So for example, I have here, this is 3 and this is 4 and this is 5. For example, the height. So let's say 3 centimeters. So the variation or the variance from each other or the pooled variation is called the sampling error because they are in the same experimental unit. They are treated the same, but however, they have the difference in their response variable and their response characteristic. So that is the sampling error. So when we say sampling error within the experimental unit, so they have received one treatment, however, they have yielded a different response. So, however, in experimental error, if they are treated the same here, for example, they are treated the same, this one, then this one, and then this one, but then they did not have this same response, then that will be called the that will be called the experimental error. Okay, so experimental error is the error among experimental units. Whereas in sampling error is the variation or it's the error among sampling units within each experimental unit. So another term is the levels or the treatment levels, the factor levels is the preset quantities of quantitative factor or treatment or categories of a factor treated under or treatment under study. For example, we have the 0 kilogram per hectare, the 30 kilogram per hectare, 60 kilogram per hectare. So these are the levels. So treatment levels or factor levels. And then layout, it refers to the final arrangement of treatment levels over the entire set of experimental units. So another example. An experiment was conducted to test the effect of different watering rates per day on a growth of a flowering plant planted in pots. So we can see here in this example that since they are in pots, they cannot be affected by each other's treatment, then you can have this, in this design, you can have the CRD or completely randomized design. So you can also use that one since they are in the different pots. So 3 flowering plant in pots were used for each rate. So of watering per day specified 0.1, 0.5, and 1.1 liter per day. So in other words, we have the 9 pots because R times T or replication times treatment is equals to our total experimental units. So we have the 9, 3 times 3 because we have the three levels and then 3 replications. So we have here the 0.1. So it might be with your layout, your arrangement, you can have the 0.1 here, 0.1 here, 0.1 here. So we have the 3 replications of watering. And then 0.5 watering rate, we also have 3 replications. And also 1 liter per day, we also have the 3 replications. So exercise 1. What is the experimental unit? So our experimental units is the water pots or the plant pots because you have poured your water in there. And then what is the sampling unit? Because we have taken the plant height. So we have the plant. And then how many treatments? So 1 treatment in 3 treatment levels because we have the 0.1, 0.5, then 1 liter per day. Then how many replications? So they are 3. And then how many experimental units? So in our discussions, we have the replications or R times treatment is equals to N or experimental units. So we have 9 experimental units. Another example. An experiment is to be conducted to determine the effects of 4 detergent brands, let's say Ariel, Breeze, Champion, and Tide, on the whiteness of clothes, measured in whiteness rating. So they have the whiteness rating. And then 16 uniformly soiled shirts, so this is our experimental units, are to be washed with 4 shirts randomly assigned to each of the 4 detergent. So we have the 4 replications and 4 treatments. So that's why we have these 16 experimental units. So what is the experimental unit? What is the sampling unit? How many treatments? How many replications? And how many experimental units? So you answer it. And so we have here another term is the precision. So the ability of a measurement to be consistently reproduced and it is measured by variance. So ways to increase precision is to increase number of samples or the skillful grouping of experimental materials or proper selection of treatments. And then definition of term for accuracy is the closeness of the observed values to the true value. It is measured by the bias difference between the average of the values and the true value. So we have also ways to increase accuracy. So it is the refinement of experimental techniques and proper selection of treatments as well. So properly selection of treatments, proper design can increase your precision and accuracy so that your study or research results would be reliable. And we have the 3 principles of experimental designs. We have replication. It is the repetition of basic experiment. So the repetition from the word itself, and it's the number of times the treatment appears in the experiment, how many times you have repeated it. And then randomization. It is a chance mechanism that gives every treatment an equal chance of being assigned to an experimental unit. So for all experimental units to be fairly treated. And then we have the error control or the local control, means by which the experimental error is controlled. So whenever I say what is your local control or how do you control the experimental error, sampling error, or in general term, you call it as the error control or local control. Okay, so what are the functions of replications? So it is to provide a measure of experimental error for you to compare that in the different experimental units, let's say pot or plot, whatever variation or variance from each other, they can give you or provide you the experimental error. So you can be compared that even if you will repeat the same experiment, given the same condition, you will have the same result. So that's the purpose of replication. To improve the precision of the experiment by reducing the standard error of treatment means, thereby increasing the precision of the estimate standard error. And of course, to increase the scope of the inference by selection and appropriate use of more experimental. And what is the functions or what are the functions of randomization? For you to have a valid measure of experimental error, to provide a random sample of observation, since probability theory carries the concepts of randomization, or in statistics, in experimental research, it is also under the experimental or the statistical probability theory. And then to eliminate systematic bias in assigning the treatments of the experimental units. And of course, to satisfy the assumptions of the independence of error, which is the assumption for analysis of variance. So that's why we have randomization. And then to minimize errors associated with experimental units that are adjacent to in space and time. So for you to ensure unbiasedness, and then to ensure that you have undergo the chance mechanism, that every sampling units have the chance to be assigned in different treatments. And then we have here the error control or the local control. So it means by which the experimental error is controlled. So all practices or techniques used to minimize the experimental error, which includes balancing, blocking, and grouping of experimental units. So what is this balancing, blocking, and grouping? So we will define it later. However, we have the common techniques of local control. So one of the techniques is use of most appropriate experimental design, especially if you have the objectives that is appropriate for for this design. Say for example, it is appropriate to use the blocking or the randomized complete block designs by using blocking, or you can just use the CRD. So that is also common techniques of local control. So it's the use of proper shape and size of experimental units. So they must be homogeneous. So use of concomitant variable and the refinement of experimental technique. So what is grouping? So placing homogeneous experimental units into groups and comparing the treatment in each group. And blocking is grouping the experimental units into blocks such that the units within a block are relatively homogeneous, just like in the RCBD. So you've used the blocking. And then grouping or balancing is the grouping, blocking, and assignment of the treatments to be in the to the experimental unit in such a way that a balanced configuration results. So it must be balanced. So through this, it makes the design more efficient, making the test of significance more sensitive or the test procedure more powerful. So by controlling some errors, so your research results would be reliable. And then we have these structures of experimental design. If I say what is treatment structure, it is consists of the set of treatment or treatments combinations or populations that the researcher has selected to study or compare. So this is your treatment structure. What is the structure of your treatments? Two are they two factors or only one? Or then the design structure. You have referred to grouping of the experimental units into homogeneous blocks or groups or blocks. So just like CRD or RCBD. So that is the under the design structure. To further explain that, we have here the treatment structure. So for example, you have the one-way treatment structure or treatment consists of levels of a single factor. Say you have the different rice variety. So only one treatment structure. So it is one-way treatment structure. So for example, and or other words, you have only used one treatment or one characteristics. Okay, but of course in a different levels. And then factorial. Factorial is treatment consists of all possible combinations of the levels of two or more factors being tested. So I will not discuss this one, but for you to be aware for your background, fractional factorial, you also have only a fraction of the full set of treatment combinations in a factorial treatment structure. So we also have nested treatment structure, but I won't discuss this fractional and the nested treatment structure. You can study it independently. So what is this one way? This is the design or the treatment structure rather, one way. So when I say treatment structure is the how you deal with your treatments. So if it's one way, remember it is only one treatment, but of course in a different levels. So comparing the bioethanol yield of three different protocols for the production of a bioethanol from sweet sorghum. So our response variable here in this example is bioethanol yield. And then this sweet sorghum produces the bioethanol. But if you've used or applied the different protocols or process, then you will also have the different bioethanol yield. So our treatment here is only the different protocols. So we have the three different protocols and our levels is three different protocols. So this is our treatment structure. It's only one. So different protocols. And then our treatment levels is three. Okay. So that is one of the example of one-way or one-way treatment structure. So if I say factorial, if I would ask you what is your treatment structure, you would answer either one way or factorial. And factorial could be 2 factorial, 3, 4, so many treatments. So for example, studying the temperature, that is another treatment in a different temperature, and concentration of a reagent on the yield of a chemical reaction. So our treatments here are the combinations of the temperature and the concentration. So that is factorial treatment structure. Specifically, the 2 factorial treatment structure. So how about the design structure? If I would ask you, what is your design structure? You will answer like this: CRD, RCBD, LSD, split plot, strip plot, or strip split design. So we'll only talk about the 1 and 2, the CRD and RCBD. So now I believe that you have understood what are the treatment structure and the design structure. If I will ask you treatment structure, you can answer me one way or factorial. So depending on how many treatments do you have. And then the design structure is your layout. Okay. So to end, here's a reminder. Commit your works to the Lord and your plans will be established. Proverbs 16 verse 3. Even in our experimental research, you have plans. So in life, we also, if we plan to be Christ-like, we also have to evaluate if we are following the right procedure, the right process in achieving that Christ-like character. So, reminder, commit your works to the Lord and your plans will be established. If in our research we have the plans, how much more our plans to be in heaven. So God bless you and don't forget to pray before you study. God bless. See you in the next topic.